Welcome to the deep dive. Today we're going to be looking at some pretty cool research actually about NMDA receptors or NMDAs. You ever heard of those?
I have.
Okay, great. So they're these tiny proteins in our brains, and they are super important for learning and memory.
Right.
And this deep dive is all about a recent study that was published in Human Molecular Genetics, and it's using like cutting-edge technology to try and predict the effects of these tiny genetic variations in NMDARs.
Right.
So we'll try to break down the science in a way that's easy to understand for our listeners, and then we'll kind of talk about what it all means. But to help us navigate this topic, we have an expert in genetics and brain function here with us today.
Hi. Thanks for having me.
So to kick things off, can you tell me what exactly are these NMDA receptors?
So imagine our brains are like this complex network of roads, and the neurons are like cars traveling on those roads.
Okay.
NMDARs are like traffic lights at intersections controlling the flow of information between those cars or neurons.
Okay, so they're like gatekeepers for signals in the brain. That makes sense. The study we are diving into today is all about tiny genetic variations in these NMDRs. Why should we even care about these variations?
Well, even the smallest changes to these NMDRs can have a big impact on how our brains function.
I see.
Think of it like this. If you have a faulty traffic light, it can cause chaos and even accidents.
Yeah.
Similarly, variations in NMDRs have been linked to a whole range of conditions like epilepsy, developmental disorders, and even some psychiatric conditions.
Wow, that's pretty serious stuff, it sounds like. Figuring out what these variations do is really important.
Absolutely, and that's exactly what the researchers in this study set out to do. They wanted to see if they could predict whether a specific variation in an NMDR would be harmful, or what we call pathogenic, or if it wouldn't really have any effect, what we call benign.
So how do they go about doing that? It sounds like trying to find a needle in a haystack.
It's definitely a challenge, but the researchers came up with a really clever approach. They used the 3D structure of the NMDAR protein and mapped out where these variations were located.
Yeah.
It's like creating a detailed map of our traffic intersection and pinpointing exactly where the problem is.
Okay, so they focus on location, but why is location so important?
Remember how I said NMDRs are like traffic lights controlling the flow of signals? Well, for them to work properly, they need to bind to specific molecules like glutamate and glycine. Think of these molecules as the keys that unlock the traffic light and allow signals to flow through.
Right, the signals that allow the cars or neurons to keep moving.
Exactly. Now, the researchers found that variations located closer to these binding sites, where those keys attach, were much more likely to mess up the NMDR's ability to function properly.
Ah, so it's all about location, location, location. If the variation is close to where those crucial molecules bind, it's more likely to cause problems. Did their hunch pay off? Did they find that to be true?
They did. Their analysis revealed that variations closer to these binding sites were indeed much more likely to be pathogenic, meaning they could disrupt the NMDAR's ability to do its job. It was a big aha moment for the researchers.
That's really interesting. So by looking at the location of these variations on the NMDAR, they could start to predict whether they would be harmful or not. That's amazing.
It is, but the researchers didn't stop there. They took their analysis a step further and used a really cool technology called machine learning to try and make even more accurate predictions.
Hold on, machine learning. I keep hearing that term everywhere, but to be honest, I'm still a bit fuzzy on how it works.
Yeah.
Can you break it down for me in a way that even I can understand?
Of course, imagine you're teaching a dog a new trick.
Okay.
You show them what to do, reward them when they get it right, and correct them when they make a mistake. Machine learning is kind of similar. You feed a computer program a ton of data, in this case, information about known NMDAR variations and whether they were harmful or not, and it learns to recognize patterns and make predictions based on that data.
So they basically showed this program a bunch of examples of good and bad NMDAR variations, and it learned to spot the difference.
Exactly. The researchers trained a machine learning program using data from previously studied NMDAR variations and their effects. They basically taught the program to recognize the patterns that link a variation's location to its potential to cause harm.
So what happened? How good was this computer program at predicting whether a variation was harmful?
It was surprisingly good. The program correctly predicted the effect of the NMDR variations with a high degree of accuracy. This is a big deal because it suggests that we might be able to use machine learning to quickly and accurately identify which NMDR variations are likely to cause problems.
This is incredibly fascinating. By simply looking at the location of variations in NMDRs, scientists were able to get a good idea of whether those variations would be harmful, and then using a machine learning program, they were able to take those predictions to the next level. This all sounds super promising.
It opens up so many possibilities for diagnosing and treating conditions related to NMDRs. But there's even more to this research than just predicting whether a variation is harmful or not. The researchers also wanted to understand how these variations were actually affecting the NMDRs.
Okay, I'm intrigued. So we know that some variations can mess up NMDRs, but are there different ways they can mess things up?
Exactly, and that's what we'll discuss in the next part of our deep dive.
All right, so before we dig into how these NMDR variations can mess things up, can we just recap what we've learned so far? I want to make sure I'm following along.
Of course, we've established that NMDRs are these vital proteins that control the flow of signals between neurons, and even tiny variations in their genetic blueprint can have significant consequences for brain function, potentially leading to various neurological and psychiatric conditions.
Right, like those faulty traffic lights causing chaos in our brain's intricate road network.
Exactly. And what's remarkable is that this study showed that just by looking at the location of these variations on the 3D structure of the NMDAR, protein researchers could get a good idea of whether they'd be harmful or not. They even used a clever machine learning program to make those predictions even more accurate.
Okay, I'm with you so far, but now I'm really curious about what you mentioned earlier, how there are different ways these variations can actually mess things up. Like, not all traffic light malfunctions are created equal, right?
Precisely. It's not just a matter of whether a variation is harmful or not, it's also about how it's causing problems. Think of it this way. One traffic light might be stuck on green, causing accidents because cars keep going when they shouldn't. Another might be stuck on red, creating a massive traffic jam because nothing can move. Both are malfunctions, but they lead to completely different problems requiring different solutions.
Okay, I get that analogy.
Yeah.
So you saying some NMDAR variations might make them too active while others make them sluggish?
Exactly. Some variations might make the NMDARs overactive, sending too many signals between neurons, like a constantly flashing green light. This could lead to conditions where the brain is overstimulated, potentially contributing to things like seizures or anxiety. On the other hand, some variations might make the NMDRs underperform like a dimmer switch stuck on low, hindering the flow of signals. This could result in problems with learning, memory, and other cognitive functions.
Wow, that makes total sense.
Yeah.
It's not just about whether a variation is bad or good. It's about how it's tweaking the NMDR's activity, either ramping it up or slowing it down.
You got it. And the incredible thing is that the researchers in this study were able to go beyond simply predicting whether a variation was harmful. They could actually predict how it would affect the NMDR's activity, whether it would make it more or less active.
Wait, seriously? They could predict not just if a variation would cause trouble, but how it would cause trouble? That's like some next level detective work.
Yeah.
How do they even do that?
Remember how we talked about those crucial binding sites where molecules like glutamate and glycine attach to the NMDR?
Yeah, the keys that unlock the signal flow.
Right. Well, the researchers found that variations located near these binding sites, but not directly on them, could actually change how easily those keys could fit and unlock the NMDAR.
So it's like if you slightly altered the keyhole, the key might fit more loosely or more tightly, changing how easily you could unlock the door.
That's a great analogy, and depending on how those variations affect the binding of those crucial molecules, they can either enhance or hinder the NMDAR's activity.
This is blowing my mind. It's like they've given doctors a superpower, the ability to not just see if a variation is harmful, but also how it's causing problems.
Exactly. Imagine being a doctor who can look at a patient's genetic profile and say, okay, this variation is making your NMDRs overactive, so we need to use a treatment that calms things down. Or this
Mhm.
variation is causing your NMDRs to underperform, so we need to give them a boost. This research could revolutionize how we diagnose and treat NMDAR-related conditions.
It really feels like we're on the cusp of a new era of personalized medicine, especially for these complex neurological and psychiatric conditions.
It's an exciting time to be in this field, and while there's still much more research to be done, this study provides a powerful new tool for understanding and potentially treating NMDAR-related disorders.
This deep dive has been absolutely fascinating. I feel like I've learned so much already. We started by exploring what NMDARs are and why these tiny variations matter. Then we learned how scientists can predict whether a variation is harmful just by looking at its location and even use machine learning to boost those predictions. And now we've uncovered how those variations can actually mess with NMDAR activity, either making them overactive or underactive. It's incredible how much information we can glean from these tiny genetic changes. But before we get ahead of ourselves, what are your thoughts on the limitations of this research? What are the next steps?
That's a great question. This study was incredibly insightful, but it's important to remember that it's just one piece of a much larger puzzle. There's still so much we don't know about NMDRs and how they contribute to various conditions. For example, This study primarily focused on the location of the variations on the NMDR protein itself. However, there are other factors that can influence how NMDRs function in the brain.
Like what?
Well, for one, we need to consider how efficiently these NMDRs are transported to their proper locations within neurons. Imagine building a perfect traffic light, but then placing it in the middle of a field instead of at an intersection. It wouldn't be very effective, would it?
Right, so even if the NMDR itself is functioning correctly, if it's not in the right place, it won't be able to do its job properly.
Exactly. And there are other factors to consider, such as how these variations might interact with other genes or environmental factors. It's a complex interplay, and unraveling all the intricacies will require further research.
So it's a great starting point, but there's still a lot more work to be done before we can truly harness the power of this knowledge for personalized medicine.
Absolutely. But this study definitely provides a solid foundation and a powerful new lens through which to view NMDRs and their role in brain health.
Wow, that was a lot to unpack. I was still kind of reeling from the fact that these tiny genetic variations in NMTRs can have such a big impact on how our brain works, you know?
It is pretty remarkable how such small changes at the molecular level can like ripple out and affect our thoughts and feelings, even our behaviors.
It's like a domino effect, isn't it? You know,
Yeah,
one
yeah.
tiny little nudge can set off this whole chain reaction.
Yeah,
Wow.
exactly. And what's really interesting about this research is that it's not just about finding these variations, it's about understanding how they work, how they affect that balance of brain activity, and then how we can use that to, develop some targeted therapy.
Yeah, speaking of targeted therapies, this research seems like a huge step toward personalized medicine for, conditions related to NMDRs.
Absolutely. Imagine like a future where we can analyze someone's genes, pinpoint any variations in their NMDRs, and then come up with a treatment plan just for, could it be like having a custom repair manual for each person's brain?
It's mind-blowing. It really feels like we're on the edge of like a whole new way of
Mhm.
thinking about treating, you know, neurological and psychiatric disorders.
I totally agree. It's a super exciting time to be working in this field. We're finally starting to understand the brain and how it works and use that to help people who are struggling.
This deep dive has been quite the journey. We started out by exploring what NMDRs are and why these variations matter. Then we learned how scientists can predict if a variation is harmful just by looking at where it is and even use machine learning to, make those predictions even better. And then we just finished talking about how those variations can actually change NMDR activity, making them overactive or underactive.
Yeah, it's been like a whirlwind tour of science.
And I think it's safe to say that this research could change lives.
Oh, definitely. As we learn more about NMDRs, we'll be able to diagnose, treat, and even prevent a whole range of conditions.
It's like opening up this whole new world of medicine.
Exactly. And it's a world that's full of possibilities for improving, people's health and well-being.
Well, listeners, I think that's about all the time we have for today's deep dive. We've covered a lot, but I hope you learned something new about these fascinating proteins called NMDRs and how they could lead to personalized medicine. A huge thanks to our expert for, sharing their knowledge and experience with us. It was great having you.
Thank you. It was fun to talk about this research.
And to our listeners, thank you for joining us on this deep dive. We'll be back soon with another one into the world of science and technology. But until then, keep those brains buzzing.